carelessly during fishing and transport may also facilitate parasitic infection. The prevalence of fish diseases is very much dependent on the intensity of stocking. So when a farmer decides to raise the stocking rate, he not only has to provide extra food, but also has to take special care to prevent and cure outbreaks of diseases. So diseases are more common in freshwater environments, as it has been found that susceptible freshwater fish are significantly free from disease when grown in slightly brackish water. So properly managed ponds usually remain free from diseases. Carelessness in stocking and feeding may result in serious parasitism and mortality. Prevention is better than cure. Care should be taken to prevent parasites gaining access to the culture ponds from any nearby infected source. Now, how do we know that the fish is having some disease? What are the preliminary observations we can make so that we can come to one conclusion that the fish is, we can diagnose the disease and we can give the treatment and all that. So fishes are poikilotherms, hence the environmental impact is more in fishes when compared to warm-blooded animals. The following aspects are useful for the identification of diseased fishes. 1. Disease can be diagnosed only in freshly killed fishes and live fishes. If it is late after the death of fish, diagnosis is very difficult due to the chemical changes in the body and normal temperatures. Slime production is more in diseased fishes. After death, it settles on the bottom of pond, then floats on surface of water due to the gases produced by chemical changes that occurs in the body. Mucus samples should be collected from body surface and gills should be examined under microscope. Then change of body coloration, abnormal behavior of the fish. Then examine the external features and later internal examination also should be done. Examine the size, color, and shape of the internal organs like liver, kidney, and spleen. Also examine the fluid accumulation, hemorrhages, and inflammations in the body cavity of fish. Take out the samples from vital organs and go for bacteriology, virology, and histological studies. Examine for tumors or swelling in the body of the fish. Let us discuss about a few viral diseases. So viruses are transmitted from one host to the other through a structure called virion. Viruses are classified mainly based on external structure, shape, size, capsid structure, RNA, and DNA nucleic acids. Viruses cause disease by weakening the host tissue or by forming tumors in the host tissues. There is no treatment for viral diseases. Only prophylactic measures have to be taken. Now, the first disease name is lymphocystis. Woodcock identified this disease in fishes, marine, freshwater, and aquarium fishes. So these are the susceptible to this disease. Tumor formation is the important character of this viral disease. Trials external lesions raised and made up of granular nodular tissue, which is composed of mainly many greatly enlarged host cells. Matured lesions may become slightly hemorrhagic. Within 6 to 15 days of infection, the tumors grow to 1,000 times. It causes a lot of damage in the Baltimore Sea area in America. Next is viral hemorrhagic septicemia. So in short, we call it as VHS, viral hemorrhagic septicemia. This disease is caused by an unequal shaped fish virus with RNA. So this disease occurs in mostly in salmon fishes. Transmission occurs through the water by a flagellate. This disease is also called as infectious kidney swelling and liver degeneration in German and pernicious anemia. Infectious or enterohepatic renal syndrome in France. The symptoms are kidney swelling, reduced appetite, obvious distress, erratic spiral swimming, multiple hemorrhages in skeletal muscles, change in body color, reddish fins. The only control measure is prevention. So these are the symptoms that we see. What are the symptoms? Kidney swelling in kidneys, appetite is reduced, and erratic spiral swimming will be there. That means they swim around in a circular manner. Multiple hemorrhages in skeletal muscles, and the body color also changes. And the fin color also changes into red color. So these are the symptoms of viral hemorrhagic septicemia. Next is infectious pancreatic necrosis. In short, we call it as HPN, infectious pancreatic necrosis. This disease is found in trouts. So it causes high mortality of fry, fingerlings, and occasionally larger fish also. The symptoms are darkening distension and at times hemorrhages in ventral areas, including base of fin. There is pronounced pancreatic necrosis. Chlorine at 200 ppm is effective for treatment. Next, infective hemopoietic necrosis. In short, we call it as IHN, infective hemopoietic necrosis. This was observed for the first time in trouts in British Columbia in 1967. Necrosis it is observed in the hemopoietic tissue of kidney in infected fish. This disease occurs more in fry and fingerlings and occasionally in adults. The symptoms are pale gills, reddish fins, black coloration of the body, abdomen swelling, and huge mortality also. The symptoms are clear in 12 to 45 days after the entry of virus into the host body. Next is Chinook disease. So a small site virus is responsible for this disease in Chinook salmon. The symptoms are exothalamus, distended abdomen, dull red areas on the dorsal surface, anterior to dorsal fin. The liver, spleen, kidney, gills, and heart are pale. So this disease is transmitted by the egg from the carrier female, and there is no treatment for this disease. Next is channel catfish virus disease. This disease occurs in fingerling of catfish, Lacterolus punctatus. The symptoms are that the fish show abnormal swimming and rotating, hemorrhagic areas on fins and abdomen, and fluid accumulation in abdomen and pale gills also you can see. There is no treatment for this disease. Destruction of infected fish may prevent spread of this disease. See, these are the various viruses that affect the fish. You can, if you see the pictures, you can understand. These are channel catfish, how once they get attacked with this virus, how they look, the diseased fish. This is the fish attacked by hemorrhagic septicemia, and this is the fish attacked by lymphocytosis. Now let us discuss about few bacterial diseases in fishes. The first one is furunculosis. So bacteria are normally responsible for many fatal diseases in fishes. Like there are many diseases we're going to discuss on furunculosis, columnaris, fin or tail rot, vibriosis, dropsy, cotton mouth disease, and tuberculosis. So if you see furunculosis, it is caused by Aeromonas salmonicida in mostly in salmon fishes. It is a non-motile gram-negative bacterium. It frequently appears to infect fishes living in the dirty waters containing a large amount of decaying matter. It is also observed in few other fishes. The first symptom of this disease is appearance of boil-like lesions. If you observe the fish, how the affected fish looks like, you can see the lesions on the body surface. Other symptoms are bloodshot fins, blood discharge from the vent, hemorrhages in muscles and other tissues, and necrosis of the kidney. Bursting of boils allow the spread of this disease among other fishes and also offer suitable areas for fungus growth. In acute forms, it is systemic bacterial infection, a septicemia with bacteria present in the blood, all tissues and lesions. Fishes severely get infected with the bacteria, they die in good number. Now, how it has to be treated? Remove the severely infected fishes from the pond and supply food containing antibiotics like sulfonamides or nitrofurans. Sulfonamides like sulfadiazine or sulfaguanidine are given orally with food at the rate of 22 grams per 100 kgs of fish per day. And other antibiotics like chromomycetin and terramycin are most effective at a dose of 5 to 7.5 grams per 100 kgs of fish per day. Disinfect the eggs with 0.015% solution of methiolate or 0.185% of acriflavin. The next disease is columnar disease. It is caused by Chondrochus columnaris and Cytophaga columnaris in many freshwater aquarium fish. It is a long, thin, flexible, gram-negative slime bacterium, myxobacterials. So this disease is often associated with low oxygen level. Initially, it is marked by appearance of grayish white or yellowish white patches on the body. The skin lesions change to ulcerations and fins may become frayed. Gill filaments are destroyed and eventually lead to death of the fish. You can observe the diagrams, the pictures if you see how the affected area looks like. So they have yellowish white patches on the body and it looks like ulcers are formed on the body and even on the fins. The fins also will become like this. And the gills are also destroyed and eventually it leads to death. Now, how to treat them? Addition of 1 ppm copper sulfate in the pond to control this disease is effective. Tetramycin administered orally with food at a rate of 3 grams per 100 pounds of fish per day for 10 days is very effective. Deep treatment in malachite green, 1 is to 15,000 for 10 to 30 seconds and 1 hour bath in 1 ppm furinase is very effective to control this disease. The next disease is fin or tail rot. Tail or fin rot disease is caused by Aeromonas salmonicid and Aeromonas liquefaciens. However, protozoans and fungi may also be involved in this. It is characterized by appearance of white lines along the margins of fins. The opacity usually progresses towards the base, eroding them and causing hemorrhage. The fin rays become brittle first and later break, leading to the complete destruction of the fins. The infection may also spread on the body surface. Fin and tail rot are associated with poor sanitary conditions in fish ponds and with water pollution in nature. When you observe the fin of the fish, the tail fin, how it is rotten and it breaks and finally it leads to the death. Now, how to treat this? The fin or tail rot may be checked at an early stage by keeping fishes in 0.5% copper sulfate solution for 2 minutes. Control may be achieved with 10 to 50 ppm tetramycin and 1 to 2 ppm of benzalkonium chloride. In severe infections, the affected parts are surgically removed and the fishes are then kept in 0.04% potassium dichromate. The next disease is vibriosis. Vibrio bacteria are the causative agents of vibriosis disease in salmon and many other fishes. This disease may occur in waters with low oxygen levels. These bacteria are small gram-negative bacilli, characteristically curved. Diseased fishes show large, bright color, bloody lesions in the skin and muscles. Hemorrhages in eyes, gills may bleed with slight pressure, and inflammation of the intestinal tract. And the treatment is sulfamethoxam at a rate of 2 gram per 100 pounds of fish per day gives good results. Terramycin at 3 to 4 grams per 100 pounds of fish per day for 10 days of also 10 days also gives satisfactory results. If you look at this picture, see how the vibriosis affected fish. It has bright colored, the lesions are bright red in color. The muscles, hemorrhages in eyes also. So that hemorrhage means there is bleeding in the eyes. Gills also may bleed. There is bleeding in the gills. So this is how the affected fish look. Next is dropsy. Pseudomonas punctata is the causative agent of this disease. It is characterized by accumulation of yellow-colored fluid inside the body cavity, protruding scales and pronounced exothalamic conditions. This is known as intestinal dropsy. In case of ulcerative dropsy, ulcers appear on the skin, deformation of backbone takes place and show abnormal jumping. This is a fatal disease in culture systems. Now, how to treat? Removal and destruction of fishes followed by draining, drying and disinfecting the pond with lime are preventive measures to control the disease. So it's a very laborious process. Since we grow these fish in culture medium, so we need to clean the entire pond, remove the fish, drain them, completely dry the pond and clean, disinfect the pond, and then you have to take the preventive measures also. Now, the infected fish may be cured with 5 ppm potassium permanganate for 2 minutes dip bath. Streptomycin and oxytetracycline give good results. The next is cottonmouth disease. The filamentous bacteria, Flexibacteria, is the causative agent of this disease. The main symptom is appearance of fungus like tuft around the mouth. This can be treated with antibiotics like 10 ppm chloramphenicol for 2 to 5 days and 0.3 ppm furanase for long-term bath. This is how the fish look, cottonmouth disease. It looks like as if the balls of cotton is present on the mouth area. Even this fish also, you can observe this. Next is tuberculosis. Mycobacterium is a disease-causing agent which is difficult to diagnose without pathological examinations. The symptoms are ulcers on body, nodules in internal organs, fins or tail rot, loss of appetite and loss of weight of fish also. Now, how it has to be cured? This can be cured with dip treatment in 1:2000 copper sulfate for 1 minute for 3 to 4 days. We need to continue this. Now, antibiotics are not successful. The fishes should be destroyed and potassium permanganate or lime should be used in the pond. This is how the diseased fish look, and this is the infected areas, and how even this, how a diseased fish look. Then the other one is bacterial gill disease. This is caused by myxobacteria in salmon fish. Many bacteria are found in swollen gill lamellae, which show proliferation of the epithelium, and symptoms are lack of appetite. This disease is transmitted through water from infected fish, from one fish to other fish. It can be treated with 1 to 2 ppm of Timsen or 1 ppm copper sulfate. If you look at these pictures, how the gill area, how the gill gets, this is why it is called bacterial gill disease. Even this picture you can see, even here. These are the infected fish. Next is saprolegnosis. Causative agents are saprolegnia species. Now, what are the species that affected? Freshwater fish, carps, and goldfish. Now, the symptoms are formation of white cottony growth on fish eggs or tissues. Fish becomes lethargic. It gets tired quickly and responds slowly to stimuli. Histopathologically, rapid dissection of epidermis with slight inflammation is also observed. And this can be treated with 1 to 3 ppm malachite green for 1 hour or 1 is to 500 formalin for 15 minutes. Next is bronchiectasis. This is also called as gill rot. This disease is caused by Branchiomyces demigrans and Branchiomyces sanguinis. It is reported to be common on cultivated fishes in ponds having abundant decaying organic matter. The tubules of fungus grow into the respiratory epithelium of the gills, causing inflammation and damage to their blood vessels. The blood supply is stopped to the infected area, and as a result of which it becomes necrotic. It can be controlled with 5% common salt for 5 to 10 minutes. See, this is how the fish gills look, the infected fish. So mainly it affects the epithelium of the gills. There is an inflammation in the gill area. Next is ichthyophanosis. It is also known as reeling disease. It is characterized by swinging movement of the infected fish. It is caused by Ichthyophonus porphyri. It enters into the host along with the food. The spores spread to the various organs and in severe cases spread out to the skin, which may rupture and become ulcerative at several places. It is extremely difficult to control this disease. The infected fishes are isolated from the stock and kept for treatment in separate ponds. Medicines like sulfamethanase, terramycin, erythromycin, and calomel are useful to treat this infected fish. If you look at the fish inside the muscles of the fish also, you can see the affected areas. So we discussed till now about viral diseases, bacterial diseases. Now let us see protozoan diseases. The first one is whirling disease. This disease is caused by a Myxosporidian protozoan, Myxosoma cerebralis, only in salmon fishes. The symptoms are pancreatic necrosis, lesions and disintegration of the cartilaginous skeletal support of the organ of equilibrium. Rapid tail changing type of whirling is often seen when the fish is frightened or trying to feed. The typical symptoms usually appear at 12 months after exposure to the disease. So if the pond contains all infected fish, it is better to destroy them by deep burial. Then the pond should be cleaned thoroughly and disinfected with calcium cyanamide, quicklime or sodium hypochlorite. Next is costiasis. So this disease is caused by a mastigophore, Costia necatrix, in mostly in culture fishes. So this is a common disease in ponds where fishes live densely in water with a low pH and poor condition food. The parasites live in large numbers on fish skin, fins, and gills. The symptoms are appearance of gray-blue film on the skin, which turns to red patches in severely affected cases. The infected fish becomes weak. Loss of appetite occurs and finally they die. They can be treated with 3% common salt for 10 minutes or 1 is to 2500 formal in solution. This is how the affected fish looks. It has patches on the skin, gray blue film on the skin, and they turn into red patches when they severely get affected. Next is ichthyotheriasis. This is caused by a ciliate Ichthyotherias multifilus, Ichthyotherius multifilus. So this disease is also called as HR white spot disease. The young parasites moving in water get attached to the skin of the fish. They grow between the epidermis and dermis and after becoming large in size fall to the bottom of the pond. Infected fish develop small white spots on the skin and the fins. These parasites attack the gills also. Fish respond by jumping in the water and rubbing their body against the water objects. Respiration gets affected and they finally die. Now, how the treatment should be given? Dip treatment in 1.5 ppm of malachite green or in 10 ppm of acriflavin gives good results. 3% salt solution, 1 is to 4000 formal in 1 is to 1 lakh quinine hydrochloride or 1 is to 5 lakh methyl blue are also useful to treat this fish. Next, helminth diseases. So there we are going to discuss about few diseases which are caused by few helminthes. First one is dactylogyrosis. The monogenic trematode Dactylogyrus, it is reported to cause serious infections in fishes. Dactylogyrus extensis, Dactylogyrus vastator, and Dactylogyrus lamellatus are found in carps. So these are found on the body, fins, and gills. The parasites start appearing in the ponds during the rains, but their prolific multiplication take place during winter, when the intensity of infection on carp fry may reach as high as 94%. The most infected size group is 61 to 100 mm, irrespective of species. Infected fishes rest near the surface of the pond margin, swim very slowly, feel suffocation. These are are more slimy, dropping and folding of fins and pale gills also we can see. Now, how it has to be treated? Alternative baths with 1 is to 2000 acetic acid and 2% sodium chloride are effective. 10 ppm of potassium bath for 1 to 2 hours and 5 ppm in the pond may give good results. Bromex 50, 0.18 ppm and Dilox that is 0.25 ppm are effective to control the disease. This is how the helminth looks. The next is gyrodactylosis. Another monotreme trematode, Gyrodactylus, also causes disease in culture ponds. This also lives on fins and on the body of the fish. The symptoms are production of more slime, damage of fins, and fading of the body color. The medicines used in control of dactylogyrosis are also effective to control this disease. Other helminthes like Diphyllobotherium, Bathriocephalus, Diplostromum, Clinostromum, and spring-headed worms, these cause diseases in fishes. Nematodes also cause diseases in fishes, of which some of the common nematodes are Philometra and Camellinus. This is how the affected fish looks, and this is the causative organism. Next, leeches diseases. Leeches belong to the Gnathodella and Rhynchodella, attack the fishes. So leeches like Pisicola, Myzodelu, and Hemiclepsis hold the skin of the fish and suck fish blood. After the blood meal, they detach themselves, leaving the wound open for secondary fungal infections. The growth of fish is affected and they become weak. A popular control method is deep treatment in 2.5% sodium chloride for 30 minutes. This helps to detach the parasite from the body of the host. Use of 1 ppm Dilox for 5 days. Remove the infected fishes from the pond for treatment and drain and disinfect the pond with the lime to destroy the eggs and adult leeches. See how the leeches catch hold of the fish and they destroy the skin of the fish. Next, let us discuss about crustacean diseases. Under that, the first one is argilosis. Argulus, it is also called as fish lice. It's a common copepod parasite in fishes. It is a large ectoparasite and can move over the body surface of the fish. Argulus puncture the skin, it punctures the skin and inject cytolytic toxin through the oral sting to feed on the blood. The feeding site becomes a wound and hemorrhagic, providing ready access to secondary infection of other parasites, bacteria, virus, and fungi. Argulus transmits dropsy in fishes. In advanced stages, fish swim erratically, show growth loss and loss of equilibrium. How to control this? To control Argulus, removal of submerged vegetation, wooden lattices placed in the pond, which serves as an artificial substrate to deposit its eggs, can be removed at intervals to kill the eggs. 500 ppm of ammonium chloride, 410 ppm of balsam, 10 ppm of DDT for 25 seconds dip, 0.25 ppm of Dilox, and 2000 ppm of Lysol for 15-second dip are effective to kill Argulus. The fish is affected with fish lice. This is how the lice looks and how they infect the fish skin. You can observe these pictures. The next is Leishmaniasis. Leishmaniasis. It is caused by a copepod parasite, Lernaceae, or anchor worm. So this is also called as Lernia or anchor worm. This disease is mostly caused by Lernae cyprinaceae. The larval stages are temporary parasites that feed on mucus and blood of fish. The adult female is a specialized fish parasite, worm-like, which burrows into the fish flesh, keeping its eggs cases protruding out of the fish body. Male larvae do not attack the fish and are not specialized for parasitic life. Early infected fish swim erratically, flashing against the sides and bottom of the ponds. Heavily infected fish swim upside down or hang vertically in the water. Only partial control of larvae is possible with chemicals because the head is buried in the fish tissues and there are no exposed respiratory organs. Hence, prevention is more effective than control. 1% common salt eliminates larvae in 3 days. 250 ppm formalin for 30 to 60 minutes. 0.2 ppm gamaxane for 72 hours. 2 ppm of Lexone, 0.1 ppm lindane for 72 hours, and 1 ppm chlorine for 3 days may give good results. Observe these slides. So Lerne or anchor worm, this is how the anchor worm looks. And these are the worms affected area of the fish body. And here this is one worm and here is one worm. Next one is Ergacillus and Salmonella. These two parasites are responsible for huge mortality of fishes in the culture systems. These two parasites are found attached to the gill filaments and feed on blood and epithelium. Later, they may also be found on the fins and body. The infection results in impaired respiration, epithelial hypertrophy, anemia, retarded growth, restlessness, and finally, death. The fish becomes susceptible to secondary infection, especially fungus. Ergacillus can be treated successfully with a combination of 0.5 ppm copper sulfate and 0.2 ppm ferric sulfate for 6 to 9 days. Salmonella can be controlled with 0.85% calcium chloride, 0.2% copper sulfate, 1.7% magnesium sulfate, 0.2% potassium chloride, and 1.2% sodium chloride for 3 to 4 days. Ectheris is a common parasite attached to the gill rackers of fishes, but doesn't damage gill filaments. It can also be controlled by the above chemicals. So these are the affected gills. Next, let us discuss about few algal diseases. Cyanophyceae member, Oscillatoria, is responsible for fish mortality. So what is the causative organism name? Oscillatoria. It is found on gills and fish body in large numbers and produce toxic substances which are responsible for fish kill. Chlorella and Pharmedium also cause discomfort in fishes. Next, epizoic ulcerative syndrome. So this is the disease name. In short, we call it as EUS, that is epizoic ulcerative syndrome. It causes severe damage to India's aquaculture, especially at the moment when the Indian fisheries industry is poised for a great leap forward with a high input based high-tech production systems. So widespread outbreaks of this disease occur suddenly and often cause mass mortality in freshwater and brackish water fishes, causing anxiety and tremendous concern. Although the disease has been known in the Asia Pacific region since 70s, it appeared for the first time in India in 1988 and has now covered almost the entire length and breadth of the country. If you look at the fish, how the body is completely damaged by this algae. This is one of the algal disease, episodic ulcerative syndrome. All over the body of the fish, we can see the ulcers, the wounds, the deep wounds. Next, let us discuss about few environmental diseases. Now, what are these environmental diseases? These environmental diseases belong to non-parasitic diseases. The environment in which the fish live and grow plays an important role for fish health. Any deterioration in the environmental qualities often creates stress to fish and favor multiplication of the pathogens. Though the fish has defensive mechanism against pathogens in the form of scales, epithelial cells, acid and alkali media of alimentary canal, which offers resistance to pathogens, and filially the defense mechanisms regulated by immune system and phage cells. The pathogens predominate and diseases occur in fish farming systems. So these are not caused by the parasites or what. That is why we call them as non-parasitic diseases. Stress response from the environment leads to fish mortality in extreme cases. At lethal level, there may be several other responses like changes in fish behavior, reduced growth, food conversion efficiency, reduced reproductive potential, reduced tolerance to disease, and reduced ability to tolerate further stress. Now, how to diagnose these diseases? What are the symptoms what we see? How can we diagnose these are? First is depletion of oxygen. The mouth remains open. Gills look pale with wide operculum. Bigger fishes die first. Excess carbon dioxide. So if carbon dioxide is in excess level, what happens? Excessive secretion of mucus or high pH level in pond by epithelial cells. So if oxygen levels are low, what are the symptoms? Mouth remains open, gills look pale, and operculum is wide open. If the carbon dioxide levels are more, excessive secretion of mucus or high pH level in pond by epithelial cells. Now, what happens if nitrogenous waters and ammonia gets accumulated in the lakes or ponds? Gills look dark red due to formation of methemoglobin, a combination of nitrogen and hemoglobin. Then, supersaturation of oxygen or nitrogen. Due to this, what happens? Accumulation of gas bubbles within the body cavity of fish spawn. Now, excess of hydrogen sulfide gas and pond muck smells like rotten eggs. So what happens? The bottom dwelling fish come up to the surface and die. So if excess of hydrogen sulfide gas is present, the fish that dwell in the bottom level, they come to the surface and die. Organic pollution. Dropping of pectoral fins in case of organophosphorus pesticide. Oozing of blood from eyes in some cases. So this is called as organic pollution. So what happens because of this? They drop the pectoral fins because of this pesticide, organophosphorus pesticide. And they also ooze out blood from eyes. Next, algal toxicosis. Algal bloom may appear in ponds due to accumulation of plenty of organic matter. or due to excessive chemical fertilizers also. Toxins released by blue-green algae like microcystic, anabina, and alphanizomenon kill other phytoplankton and cause surfacing of fish stock. Persistence of the bloom will cause toxicosis for the fish stock, showing symptoms like convulsions leading to death. Next, what happens if the temperature of the water is high? The fish on crossing tolerance limit shows the alarm syndrome initially, that is, coming up to the surface, splashing water, and finally exhausted and swimming to the bottom. Indian minor carps die when the temperature is 39 degrees centigrade, and air breathing catfishes get exhausted at 42 degrees centigrade. Next is eutrophication. Water body looks like pea soup green color due to the bloom of blue-green algae. So there is a lot of growth of blue-green algae and it covers the water. So this is called as eutrophication. The fish die of suffocation. Next, how to prevent these environmental diseases? Proper sanitation by removing muck from pond bottom regularly and exposing the bottom soil to the sun. During summer months, when water level in perennial pond remains at its lowest, lime and potassium permanganate can be used in maintaining sanitation. Liming of ponds has become a must in maintaining sanitation in nursery, rearing and stock ponds. Through restricted use of manure, fertilizers, and fish feed, both primary producer, that is algae, and primary consumer, that is zooplankton, need to be kept under control, or else the supersaturation or depletion of oxygen will create problems. Acidosis and alkalosis. What happens? What is acidosis? Aquatic plants present in high densities liberate enormous quantities of oxygen during photosynthesis, which is responsible for the formation of insoluble calcium carbonate from calcium bicarbonate, followed by the formation of calcium oxide with the elimination of carbon dioxide. So this phenomenon is known as alkalosis. So what is alkalosis? Excessive production of carbon dioxide. So this phenomenon is known as alkalosis. Excessive alkaline condition leads to the corrosion of bronchial epithelium and fins. Now, how it can be prevented? By buffering the medium by means of suitable calcification. Excessive plant growth in ponds should also be avoided. The lethal acid and alkaline ranges are less than 4.8 and in trout less than 5 and more than 10.8 in carbs and less than 4 and more than 9.2 in purchase respectively. Next is gas bubble disease. When nitrogen of the water is higher than 25% saturation due to rapid temperature change, gas bubble disease may result in, and fish fry particularly die in large numbers. Fish affected by this disease often swim at an angle of 45 degrees with their head pointing down. Other symptoms are the presence of bubbles beneath the skin, fins, around eyes, in the stomach, and intestine, or in blood capillaries also. In such conditions, water should be well agitated to bring down the nitrogen saturation below 110 percentage. So if it is 125% is more than that, it causes all this. And if we need to bring it down to below 110 percentage. Or affected fish should be transferred to other ponds. Besides nitrogen, supersaturated levels of oxygen, it should be more than 350% air saturation have also been reported to cause gas bubble disease in fishes. So air saturation also should, if it is more than 355, 350% also, it causes this gas bubble disease in fishes. Next, so after environmental diseases, now nutritional diseases. So what are these nutritional diseases? Nutritional disorders are caused by an excess of nutrient or nutritional deficiency in fish that is more than the normal requirements. Lipids, carbohydrates, proteins, vitamins, and mineral salts are also only a few of the essential nutrients for fish growth. Reduced fertility, sluggish growth, poor appetite, increase disease susceptibility, morbidity with clinical symptoms, and pathological abnormalities are some of the symptoms of nutrient deficiency. Now, the last topic is health management. So, how to maintain the health of the fish? Under this, the first one is prevention of fish disease. So, what are the general prevention methods that needs to be followed? First, selection of healthy fish seed. So, the seed that we select should be healthy. Proper density and rational culture. Careful management. Qualitatively uniform ration and fresh food should be supplied. And the water quality should be good. And prevention of fish body from injuries. Next, abolishing pathogens and control of spreading. So how to abolish the pathogens, and if they are there also, how to control the spreading of these pathogens. Thorough pond cleaning and disinfection should be followed. Disinfection of appliances. What are the appliances, that nets, and what are the fishing material that we use, rods, fishing rods and all, that should be timely, they should be disinfected. And disinfection of fingerlings and feeding platform also. And fish should be properly feeded. Spot removal of dead fish from the pond. So whenever we find some fish, some dead fish, immediately without any delay, we need to remove all the dead fish from the pond. Next is immunoprophylaxis and then chemotherapy. So the fish should be treated when any diseased fish are there. Timely, they should be removed and treated. Hope you all have understood all these the types of diseases and the treatment should what type of treatment we give to them. So what are the types of questions that we expect from this chapter? So these are the questions mostly asked, which you can write in 30 lines. Answer the following questions in about 30 lines each. Write about bacterial diseases of fish. Write about viral diseases of fish. Write about fungal diseases of fish. Write about protozoan diseases of fish. Write about helminth diseases of fish. Write about crustacean diseases of fish. Write about epizoic ulcerative syndrome of fish, that is EUS. And write in detail about health management. Write in detail about environmental diseases of fish. Or write about nutritional deficiency diseases of fish. These are the long answer questions. And some of the short answer questions that we can expect are write any two viral diseases of fishes. Since we discussed so many diseases, so many viral diseases, any two you can write. Write furunculosis diseases of fishes. Write about tail or fin rot disease. Write any two fungal diseases. Write about any two protozoan diseases. Write about dactylogyrosis disease. Or write about Lerniasis disease. Write about Ergacillus and Salmonella disease. Write about the preventive measures of environmental diseases of fish. any of these questions are asked, you can easily write by, uh, you can easily learn by seeing the slides. In every slide, each and every disease is mentioned with how it is caused, what is the causative organism, how the treatment should be given also. If each slide can carry, you'll get the proper information in this. I just would like to show all the slides once. This is Unit 12, fish diseases. This BSc second year. How to observe the fish, whether they are having any disease or not. These are the preliminary observation points. Almost 11 points are there. These, if you can remember all these points, it's very good. Then these are the few viral diseases: lymphocystis, viral hemorrhagic septicemia, infectious pancreatic necrosis, infective hemopoietic necrosis, Chinook disease, channel catfish, viral disease. So these are the viral diseases, what we have seen now. And these are the bacterial diseases: furunculosis, how the treatment should be given for furunculosis disease. And this is columnar disease, the symptoms and the treatment also. This is fin or tail rot disease. You can see symptoms, causative organism, and the type of treatment that we can give. Then vibriosis, the causative organism, the symptoms, and the type of treatment that we give. This is dropsy disease, the causative organism, the symptoms, and the type of treatment. Cottonmouth disease, symptoms, and the treatment that we give. Tuberculosis, causative organism, symptoms, and the treatment. Bacterial gill disease, causative organism, symptoms, and the treatment. Saprolegnosis, the symptoms, causative organism, and the treatment. Bronchiectasis, this is the symptom, the causative organism, and the treatment. Ichthyophanosis, the causative organism, treatment, and the symptoms are also given. Then few protozoan diseases: whirling disease, causative organism, symptom, and the treatment. Then costiasis, causative organism, symptom, and the treatment. Ichthyotheriasis, symptoms, and the treatment. Now helminthic diseases: dactylogyrosis, the causative organism, symptoms, and the type of treatment that we give. This is the type of treatment. And then gyrodactylosis, symptoms, causative organism, and the treatment. Next, leeches diseases, how the leeches, what type of diseases the leeches cause, how to get rid of those leeches from the pond. All the prevention methods are also mentioned. Next, coming to crustacean diseases, what are the types of diseases, how they should be, this how the argilosis disease can be cured, the symptoms and the treatment. Then Lerniasis disease, the symptoms, the causative organism, and the treatment. This is also called as anchor worm. Then Ergacillus and Salmonella, the symptoms and the treatment. Then coming to algal diseases, the symptoms and the treatment. Epizoic ulcerative, just now we saw one question explained about epizoic ulcerative syndrome. So we can write the causative organism and symptoms and the treatment also. Next, coming to environmental diseases, what are environmental diseases, how they're going to affect the fish. So how do we diagnose the disease also? So these are the points on the basis of what we diagnose those diseases. How to prevent the environmental diseases: acidosis, alkalosis, how the gas bubble disease is cured, controlled. Then coming to nutritional diseases, what are nutritional diseases and few types of diseases. Now health management, how to provide proper health to the fish, what are the precautionary methods that we need to take. So these are the type of questions that we can expect in the exam, students. Hope you all have understood. Thank you. Sir, I'm ending the session, sir. Thank you.